The following presentation is part of the Technology and the Corporation Conference Series, sponsored by MIT's Industrial Liaison Program.
Bom dia e obrigado pela oportunidade falar com os senhores e as senhoras, and thank you for having this chance. So I'd like to tell you today about a different aspect of challenges in utilizing biomass and opportunities for using biological conversions, particularly in the materials realm. Professor Stephanopoulos gave a fantastic introduction to many of the things that we can do in terms of making chemicals with biomass. And as a person whose research focuses on polymers, I'm inspired by the fact that many of the chemicals are targeted at making a polymeric material. As we look forward at new opportunities in innovation and technology, many of the opportunities to address human needs are critically enabled by the development and engineering of new materials. And I show here four such opportunities. One is transportation, where the huge advances in the properties of composites have led to the production of lighter, more fuel-efficient aircraft, right, addressing both needs in energy and needs in transportation in our societies. In addition, we think about growing biomass for new raw materials with improved sustainability, as Professor Stephanopoulos discussed. And in particular, this is an issue for things like recycling, accumulation of trash and waste in our society, and for sourcing of materials without relying on non-renewable feedstocks. So how can we improve the quality of our construction? How can we improve the quality of our disposable goods? In particular, things like food packaging, right, which are used to keep us safe but result in the accumulation of large quantities of waste. As a society, we also now have very ubiquitous access to information, as was mentioned in some of the discussions yesterday. But this ubiquitous access to information is essentially mediated by physical materials, right? We need displays in particular that transmit information to us, and these displays are largely becoming made of new organic soft materials such as organic LEDs that can provide higher performance properties and allow us to interface between the digital world and the human world. And then finally, as the world population grows, we have an increasing number of people who have basic needs of food, water, and healthcare, and also an increasingly aging population in many countries that changes the need for healthcare, in particular in this case. And these demand new material solutions that can provide for these needs of a growing population. So nature has, of course, evolved over many, many years to come up with very creative material solutions adapted to many different niches that can inspire us as engineers to create the next generation of materials. And so I show on this slide 5 different examples. Ultra-strong fibers in spider silk, which have a strength greater than steel by unit weight and have attracted a huge amount of interest as very, very strong fiber technology. In this dragonfly wing, at the base of the wing is a hinge made of a protein known as resin. And this hinge has to move as the wing flaps, right, many millions of times over the life of the insect without fatiguing, right, because you don't want the hinge to break during flight. And so this is a very resilient material that can essentially undergo cyclical mechanical deformation at a very, very high rate many, many times without being destroyed. Mussels have developed an adhesive to stick to rocks, inorganic surfaces, and organic surfaces underwater very, very effectively, right, in the tidal zone where there's a great deal of hydrodynamic force. And so this mussel provides an adhesive that can do something that's very difficult for us to do as humans with our current synthetic materials. Biology has also developed tough composites. This is an SEM micrograph from an abalone shell, where you have alternating inorganic and organic layers, and this gives a very, very high toughness to the shell, inspiring some of the work in composites. And then finally, nature has developed, of course, catalysts and photocatalysts that are responsible for a very large fraction of the carbon fixation in the biosphere and for many of the other chemical transformations that we currently rely on as our feedstocks today. So the question I want to pose to you is, not only can we take inspiration from what biology does, how biology builds its materials to design our next generation of materials, but can we also adapt the methods that nature is using to design and construct its materials, to design and construct our own materials? And if we do this, how might this enhance our capabilities, or how might this change the way that we can operate our sort of materials ecosystem? Right? So can we essentially use a biological process to make things that we need in our everyday life? can we make adhesives, hydrogels, elastomers and ionomers, and even make catalysts, all through bioengineering in natural systems. And so these are all photos from things that are going on at MIT and, of course, much more broadly throughout the scientific community. And so I think the answer is generally that yes, this technology has some potential, but it requires a change in the way we think and a very critical questioning of where specifically are the opportunities where these sort of biotechnological materials and biotechnological transformations might have a competitive advantage over what we can do from traditional polymeric materials, right? So where might they be better and how might they compete? And I think in terms of paradigm change, I think traditionally we think of polymer science as essentially a chemical science, right? Divorced in almost all respects from biology. And it's basically related most closely to organic chemistry. And I think that now, as biotechnology has advanced, right, biotechnology, thinking of things like cellulose and things like lignin and things like protein, these are all polymeric materials at heart. And so there is a much closer relationship between polymer science and biotechnology and biochemistry than there has been in the past. I think joining this relationship together, in addition with the advances that we've benefited from in the past decades from nanotechnology, with the new metrology and new science at small length scales, gives us the opportunity to discover new materials at the interface between this traditional physical science and the emerging and very rapidly developed biological sciences, which now offer, I think, new technologies and new capabilities that can be brought across into the materials realm. So this has been practiced and attracting a lot of attention in medical research historically, right? Because it's very important when interfacing things with the human body to consider their biochemistry and the way that they would work in a biological system. But I want to show you several examples today that could potentially go beyond just medical research. They could impact medical research, but also go beyond into other sectors where you can potentially have an impact with this sort of fusion between biotechnology and material science, right? So in asking the question, where will our materials come from, and when might it be possible to derive a material from a biotechnological source, right? I think we always ask ourselves, where would a material derived from biotech have a competitive advantage over a synthetic material, right? So what do biological systems do exceptionally well where we can design materials that will be very, very competitive with existing technology? And I want to show you 3 examples of areas where I think that biotechnological materials potentially have a competitive advantage. The first one is that biology is very, very good, as Professor Stefanopoulos highlighted, at chemical transformations, very selective chemical transformations. And so biocatalysis, of course, has established itself as an industry. And I think that looking at materials engineering in this realm can potentially have very large impact. I also want to discuss with you one simple example, just showing how when we build materials from biotechnology, we can think not of replicating the properties of an existing material, but of going fundamentally beyond in the capability, building sort of toys that we never dreamed we would have access to before in the material science realm. And then finally, I want to discuss examples of where we can take materials that for one reason or another are popular but not good materials and replace them with things that are better. So can we do things that are more sustainable? Can we do things that are less toxic? And can we do things that will potentially move us out in front of emerging regulatory trends that may change the business landscape for certain materials? For example, there have been changes in the not too distant past affecting polycarbonates and affecting fluoropolymers, right? And there are other polymer materials that are potentially in this sort of challenge. All right, so starting out, I want to talk to you about protein materials and catalysis, right? Biocatalysis is a huge industry. The synthesis of chemicals and pharmaceuticals in many processes relies on biocatalysis. So things like drug intermediates, sugar isomerization, and not too long ago, acrylamide synthesis process was commercialized that's based on biocatalytic conversion of cyanoacrylate into acrylamide. There's also these sort of biocatalysis is also very popular for medical applications, where by far the largest penetrating application is the glucose sensor, right, which uses glucose oxidase as a selective detection method for glucose in the blood, relying on the very high selectivity of the biological conversion. Now, the challenge is that most of the enzymes we'd like to use for this biocatalysis exist as homogeneous catalysts in the cell. And this is very challenging from an engineering perspective because for most applications, such as a paper strip sensor or for building a chemical process, we often prefer heterogeneous catalysts due to the ability to easily separate and recycle the catalyst, which is often one of the highest cost components. Right? So we'd like to build proteins into heterogeneous catalysts, even though they exist naturally in solution. And there have been a number of methods to do this. But building a protein to a heterogeneous catalyst is a very large challenge because the total size per catalytic site of a protein is much, much larger than a transition metal catalyst. And so the challenge is, how do we achieve a sufficiently high density of protein in a heterogeneous catalyst while still maintaining the accessibility of that catalyst to diffusion and transport within the material? So how do we solve the classic chemical engineering challenge of diffusing things into a material while having a high enough density of reactive sites? Right, so we need to build a material essentially in 3D rather than grafting proteins on surfaces so that we can get a high density, but at the same time things have to be able to get into and out of the material, and we need to maintain the protein or the biocatalyst stabilized throughout the entire process so that it can maintain its activity. So we propose, why not think about self-assembly? Why not go to nanotechnology and look for solutions that you can use to put together proteins into high-density catalytic arrays? So one of the, I would say, you know, as a polymer person, one of the oldest nanotechnologies is this simple molecule here, block copolymer self-assembly, which was discovered, you know, of order 60 years ago now, right? And it's so inexpensive and so effective that it's used in shoe rubber, right? And it's used in adhesive on the back of Scotch tape, right? So this is one of the most widely penetrating examples of nanotechnology that operates at an extremely low and easily commercializable cost. So what if I imagine my enzyme or other protein of interest, and I could link to this something else, some other polymer, for example, that would make this look like a block copolymer, right? Then maybe I could self-assemble this into this sort of nanostructured array that I showed schematically on the last slide. And so we've developed ways to do this, both chemically and with biological process, by essentially adding either a polymer or adding a sort of protein tail coming off the end of the enzyme of interest. And I show here a TEM micrograph where you can actually build a solid piece of plastic that's 50% enzyme by weight, right? So a huge, huge density of protein in this material. And at least in the lab, we can make this at scale. So this is synthesized in my lab. These are a gram each of solid pellets of these protein plastics that you see in the hand of one of the graduate students. And so this can be made in about a week at this scale, just with our small lab scale reactors, and that was our production a couple years ago. With our new processes, you can increase the production probably by an order of magnitude at the lab scale, and the separation is much, much faster. So we think now that we're at the point where you can make these sort of self-assembled plastics at a cost that's comparable to the cost of isolating an enzyme in an industrial enzyme isolation process. You can make all kinds of different nanostructures, looking at the fundamental material physics, things that have packed spheres or cylinders, lamellar sheets, perforated lamellae. We've got bicontinuous gyroid phases now, and so you can access all this different range of morphologies that might give you different properties. In addition, you can maintain almost all of the protein in its active form. Right, so traditionally, when you process and cast a protein into a plastic, it loses a significant amount of its activity. With this sort of method, because of the gentle self-assembly, it maintains a very high activity. And what we show here is that with the addition of small fractions of additives, in the pharmaceutical industry, it would be called an osmolyte. In the polymer industry, we'd call it a plasticizer. Right? But if you add a small amount of these plasticizers, you can actually maintain 100% of the function of the protein in the final material in many cases. And so you can really retain almost everything in its original functional form. What we then showed was the ability to integrate these sort of proteins into industrial processing. And so we developed simple blade coating process for producing films of these materials in the range of hundreds of nanometers or microns thick. And so basically you just use a fluid and coat it by a blade coating process. So this is easily scalable up to large-scale roll-to-roll manufacturing technology. You simply immobilize this by submersion in a bath with a cross-linking agent, and this produces a permanently immobilized protein film on the surface It's lasted at least 10 days in continuous operation with no noticeable degradation. And when you compare the catalytic activity per unit protein of this material to other techniques for producing immobilized protein and surface coatings, we get about a 10-fold enhancement in the performance without an attempt to optimize, to individually optimize any of the techniques. So using each of the techniques essentially as disclosed technology, we get a huge 10-fold enhancement, and we've demonstrated the ability to recycle the catalyst at least 5 times with the model protein. So this really gives you a way to think differently about biocatalytic technology in terms of the ability to maintain very high activities, low-cost protein isolation, and easy recycling. I think these sort of technologies illustrate one of the potential advantages of biology and materials, and that's that biology offers the ability to be highly selective in the chemistry you do or highly selective in the recognition that you do. And so I think that there's a big future for new biomaterials in looking towards catalysis and looking towards applications in sensing for these reasons. And the advances in biotechnology are changing what we think of as the operating range of temperature, of chemical composition, of solvent that we could use for the application of these materials. So another example I want to talk to you about is innovation in hydrogels. And I think when you think of building a material from biotechnology, certainly building hydrogels for medical applications jumps immediately to mind. Because when you're building out of biological systems, we can naturally incorporate things that look chemically, structurally similar to what occurs in our in the body. Right, so you can imagine making things that mimic an extracellular matrix, or making things that contain a signal or a growth factor that communicates with your body to induce a certain biological response. And this is this growing field of smart biomaterials. And we've certainly done a number of these things. So Ali Kadamassani's lab has led some projects we participated in with injectable nanocomposites that can stop bleeding in 1/6 of the time that you can with no treatment. And we've also looked at new tissue adhesives that are made of the same chemical components as the extracellular matrix, so very, very gentle tissue adhesives. And you can also look at developing these components to have new sorts of performance. Like this is a hydrogel that, as far as we know, is one of the most extensible hydrogels ever discovered. It can be stretched to 3,000% of its original length without breaking. And we look at making very, very tough hydrogel materials. So in this project, we can actually use copolymer self-assembly as a means to toughen hydrogels to resist cracking and breaking, and can make essentially tissue fillers and tougheners that have a very, very high strength. What I want to tell you about in detail, though, is this idea that we can look to biology for brand new function and essentially extract it out into an engineering context as a basis for the innovation of new materials. And so one example of this, which I was introduced to by my colleague Katarina Ribek in the Department of Biological Engineering, is the nuclear pore. Right, so if you look, this is a stained image of cells, right, with a blue stained nucleus. And if you zoom in on this nucleus, I'll say these pictures aren't from the same piece of work, but I've spliced them together to schematically illustrate where we're looking. The membrane around this nucleus is full of many small pores. And these small pores are critical to your survival as a person, because these pores regulate what gets in and out of your nucleus. So basically, what is allowed to see your DNA, right? So this most precious genetic information is guarded by a hydrogel made of protein inside the pore of your nuclear membrane. Right? And this nuclear membrane is very selective. It allows less than 0.1% of all proteins in your cell to pass through, but it allows translocation per pore of thousands of molecules potentially per second. So it allows very rapid transport of only a few things. Right? The protein that makes up this pore has been isolated by biologists and cloned recombinantly in E. coli. And I show here a gel made of this. This is one example called NSP1, which is one of these nuclear pore proteins known as nuclear porins. And when the NSP1 is, so they do a test ex vivo with NSP1, where you take a molecule that is allowed to transport through the nucleus and you label it green, and you take one that should be blocked and label it red, and you can see that the green molecule goes into the gel and the red molecule is blocked. So the recombinant version shares the same function as the native version, not surprisingly. Now, so you can replicate outside the body this function. The challenge is that the recombinant version has a very, very poor expression yield. even after extensive efforts at optimization, and it's almost impossible to engineer because of its extremely complex protein sequence. All right, so what we want is a minimal tool that we can use for the design of materials that we can make in very large quantities. So what we did was looked at the native protein sequence and said, is there some minimal element from this complex unit that we can extract that will capture the function of the material? And if you use we look at this C-terminal end is believed in biology to be responsible for this high selectivity of the protein. And so we did bioinformatic analysis on the C-terminal end, and we plot a frequency map of the occurrence of all of the amino acids in this material. And what you can do then is, from this, extract two equally likely sequences that are the consensus for the functional unit of this protein. And so we did that, and we named them 1 NLP and 2 NLP for nucleoporin-like protein. And what we can do then is clone them into a full-length molecule just by repeating this same minimal unit of amino acids. And if you do that, you get something schematically that looks like this, where these red dots are meant to represent this FG domain, which is believed to be a biological binding domain contained within this functional unit. All right, so we make this material and we express it recombinantly. And when we express this material recombinantly, we get a huge increase over the recombinant natural protein, but we get no gel. Right? So the domain that we excluded from the protein is believed to be responsible for gelation. So what we do is we can add in, though, just like a polymer engineering, you can add in a cross-linking group. So now we take this and we engineer it like a polymer engineer, like a material scientist, add in a protein cross-linking group. So here we add an associating helix, but you can really add any sort of cross-linking group, and we've done several different ones. And you can now make this into a gel. Right? So we can replicate now in very high yield these gels. The proteins can be purified to be basically quantitatively pure by SDS Page, and the yield increases by 50 to 100-fold without optimization over the recombinant protein, which has already been heavily optimized. So you get huge jumps in the yield on the laboratory scale, and based on these yields, sort of just below in the range between 50 and 100 milligrams per liter, this is certainly at the point where for industrial biotech scale-up, you could expect to get a very significant yield of this protein if made and isolated. So when you do this, you actually find that this recombinant minimal version has the same behavior as the full protein. Right? So here we use GFP, and we put on GFP a binding domain, which indicates that GFP should be carried through the gel. We make M-cherry, which is missing this IBB binding domain. And then we add the protein important beta. Important beta is the nuclear transporter that pulls the proteins into the gel. And what you see is that the important beta will successfully carry the eGFP across, but the M-cherry the red protein will only penetrate by passive diffusion at a much, much slower rate. And so you can essentially borrow this biological constituent and use it to build a selective filter. Right? And so if you use your imagination then about the different possibilities, you can think now, if we could build a selective filter that can differentiate these two, what if we could now re-engineer these binding proteins, these carrier proteins, to seek any other component that you're interested in, right? So we could seek a toxin that's in your blood and try to remove it, right? We could seek a bacteria and bind to it. We could seek an analyte of interest and concentrate it by many orders of magnitude out of a complex mixture, right? And you could do multiple at once by building several different carriers that recognize different things and pull them into this central depot, right? So I think this is an example of where we can use biology to build something that's very difficult to conceive of in a synthetic material context, but potentially has huge value in the medical realm in terms of things like detoxification and in terms of analyte analysis and sensing of dilute analytes. Right? And I think there are many examples of this that have been successfully come out of biotechnology. So elastin-like polypeptide, which is a very, very stretchy protein that's been used in many cases of biomedical engineering and is currently being commercialized by several companies in the United States. Resin-like polypeptides, which were developed in Australia and the United States, which mimic that insect hinge in its properties, and many, many other examples where we can find these minimal sequences and make materials that have brand new capabilities. Right. And then finally, I want to discuss, coming more back to the topics that Professor Stephanopoulos raised, about thinking about what we can do with biomass to improve sustainability and look at new chemicals. And so this slide is from Chris Prather, a colleague of ours at MIT. And we've had lots of discussions about what can you do now in an economy where you're using biomass as a feedstock. And the thing that strikes me as a polymer scientist is that I traditionally do chemistry with CH2. Right? CH2 multiplied many times. Now, when I look at biomass feedstocks, I'm basically doing chemistry with CH2O or CH2ON, right? And so as a polymer scientist, I ask myself, if I have all these new chemicals, potentially I have new monomers and new opportunities. So not only could I make things that will, not only could I make from biomass the monomers I already use, but maybe I could make from biomass new things that are better than the monomers we had before, right? Because we developed the monomers we had before in the constrained context of making them from petroleum. But now if my constraint is lifted because I have access to new feedstocks, can I do new things that will be interesting and important by virtue of having fewer ties on my hands in terms of what I can do with my chemistry? And so one thing that's very interesting looking at polymers that I think is an area that's open for exploration is that so far a lot of the work has been focused on replacements for polyesters and essentially nitrogen-free polymers. But a lot of our most expensive, highest value performance polymers contain nitrogen. Things like polyimides, polyurethanes, aramids, right, are some of the most important performance materials today for things like clothing, coatings, automotive, construction, very large industries relying on these performance polymers. All of these need a nitrogen-containing feedstock, right? The amide bond or the urethane bond produces very critical properties in these materials where the chemistry directly translates up into performance. And so could we want to ask, could we take protein or could we take nitrogen-containing biomass feedstock and start to look at ways to improve on this aspect of materials science? So one example that inspired us very much is polyurethane. Polyurethane is made from isocyanate monomers, which is a very high volume monomer and extremely toxic, right? And so the use of isocyanates is not a favorable chemical process. Polyurethane also has a comparatively high sales price relative to many other polymers, and so it's a relatively economically attractive target. So we said to ourselves, the way polyurethanes perform is well known. They have crystalline rigid domains, and these crystalline rigid domains are surrounded and linked to soft domains, and it's this soft-hard morphology combination that gives them their performance. So can we replace the isocyanates in the hard domain with something else and make a hard domain from biotechnology that's going to be more healthy and more sustainable than isocyanate chemistry? Right, so we were actually inspired by another negative thing, which is neurodegenerative disease. Many proteins, when you have neurodegenerative disease, misfold into a highly crystalline structure. And this highly crystalline structure looks a lot like what I want my polyurethane to look like. So the idea is, can you take a protein biomass, can you misfold it into something that looks like amyloid fibril, and can you use this then as a reinforcing domain inside your polyurethane? And if you could, then you could potentially transform challenges of toxicology and challenges of human health into a positive solution for sustainability. Right? And so earlier this year, we had the first successful attempt at doing this. And so we developed a process to take biomass protein, acrylate it, and then copolymerize it with very low-cost acrylate monomers to produce essentially a thermoset resin where the protein acts as the reinforcing domain, completely replacing the isocyanate. And so we basically dissolve protein, acrylate, and then do a radical polymerization to set the resin, resulting in essentially a solid elastomeric piece of plastic, basically an elastomeric acrylate that's cross-linked now by protein and can start to mimic the properties of the polyurethane material. Right. Another interesting example, I think, in this nitrogen-containing space is looking at where biology produces proteins in very high yield and what the properties of those might be. So I assume many of you in the audience have heard of polyhydroxyalkanoates as sort of a biopolyester. It turns out there's an an analogy polymer that's based, instead of being based on hydroxy acid monomers, being based on amino acid monomers, and that's cyanophycin. Cyanophycin is used by bacteria in the same way as a nutrient storage area, and it accumulates in large granules in cyanobacteria. It's a protein formed by repeat units of 2 amino acids, where you have an aspartate backbone with mostly beta-linked arginal side groups, but also a significant fraction of lysine in many of the proteins. And so it essentially forms a zwitterionic protein complex. It can be moved into E. coli and produced in very, very high yields. So we had an undergrad working on this project, and he, under the supervision of a graduate student, and he was able to get up to 1 gram per liter yield, again, without any sort of biotech optimization. So produce very high quantities of these materials from protein. And then we were very excited to see, what can this do? Right? So this is an example of something in nature that hasn't been characterized as a material. It's just been characterized in a biotechnological context. And so we look at this, we can start to measure mechanical properties. We can think of things like derivatization in the same way you think of cellulose derivatization, things like methyl cellulose, cellulose acetate. You could think of cyanophycin acetate, methyl cyanophycin, and so we can prepare those derivatives and produce very much modified properties, turn things from hard ionomers, which is basically what this is. It forms a brittle ionomer, or you can transition into more fluid type of materials, all by changing the functionalization. So I think there are also things left still to discover in these types of materials. All right, so I want to end there, acknowledge a lot of people who've supported this research, and more importantly, the group of people who've performed this research. One of them is actually in the audience, Professor Valencia Martins, who spent some time with us and worked on the polyurethane work. Thank you. I'd welcome your questions.